and he's going to talk to us about transplants to therapeutics and the role of how to standardize the microbiome for benefiting patients. Thank you, Alex. We look forward to your talk.
Thank you so much. Thank you, Sabine, for letting me speak here. Neil has done an incredible job giving an introductory talk. I'm not sure I'm going to add necessarily that much. He almost challenged me to talk about the terminology here. And he knows I hate the term fecal transplants. That is a unfortunate, unforeseen terminology that was never intended. About a decade ago, a bunch of us got together, Larry Brand, who's been pioneering this treatment longer than I have, raised the question. We're putting fecal material into people. It's called, at the time it was called fecal bacteria therapy, but is it just bacteria? What is it that we're doing? And so collectively, based on some data that we actually produced at the time, we called it fecal microbiota transplants. It is the microbiota that we're transplanting, not feces, which is far more complex substance. But I found that trying to modify language is a hopeless crusade. And so I'm pretty close to giving up and call whatever you want. The commercial guys, of course, will never call anything fecal. They'll call it perhaps some yogurt type name, rejuvenating, rebiotic, something like that, or some letter numbering system. And that's probably more appropriate, in fact, because we're talking about drug development. And I'll mention that and I'm actually agreeing with that. This is a new frontier of pharmacology. These therapeutics are different from small molecule drugs. They're different from biologics like anti-TNF drugs or whatever we're using increasingly in different diseases. And I'll just take you through the journey that I took over the past decade. I don't have the little arrow that is supposed to advance the slides. Oh, there it is. Okay, but I still don't see the arrow. Maybe somebody advanced it for me. Okay, seems like it's working. So this was the index case that got me into the field. It was a 61-year-old woman. When she saw me in the office, she already had diarrhea for 8 months. It started after antibiotic treatment. She's been in the hospital a couple times for dehydration and received more antibiotics. She was truly miserable. She had bowel movements every 15 minutes, 24/7. She lived in a wheelchair, funny spelling there, and lost over that period of time 60 pounds of weight. And I proceeded to treat her with more antibiotics for the next 7 months. But basically, it was pretty clear she needs new microbes. And that time, it was still 2008. This was novel. And first thing I did was check with the ethics and IRB at my institution that if we're going to do something like this, what are the implications? And they said, well, it's an N of 1 kind of case. It's not really systematic research. You don't really need IRB approval. Then I meekly asked, well, can I study her samples before and after? What is it actually we're doing? And they said, you know, you should do that. And so this was, on the left was her colon, actually at the time, Pseudomembranous colitis. You see these explosions of fat on the surface. For reference, on the right, you see the normal colon with, you can see little blood vessels cruising through. That means the lining is thin enough that you're able to see that. The condition is called pseudomembranous colitis. And Neil has gone over the pathophysiology of this condition. What she was suffering from was recurrent C. difficile infection. So normally, your colon is home to a very dense microbial community, as you all know. When you take antibiotics, that community is disrupted. That's when you become vulnerable to invasion. Typically, C. difficile is acquired from the environment in the form of spores, and they're resistant to outside environment. But once they find themselves back in the intestine, which is their natural home, and the host is susceptible, then the spores germinate, and the live bacteria, which we call vegetative forms, produce toxins, and then you can see inflammation. And on the picture on the bottom, you actually see another one of our patients with pseudomembranous colitis. The host is actually trying to patch up the holes in the lining of the colon, and the neutrophils, which is the soldiers of the immune system, spill their guts and patch up the holes, and that's the pseudomembranes. And then we treat that condition with antibiotics again. Initially, it might work, but the more you try this approach for an antibiotic-induced disease, treating with antibiotics, it turns into this recurrent infection syndrome, and you cannot get rid of it. The reason is the antibiotics cannot kill the spores, but at the same time, they're killing normal microbes in your colon and make you increasingly more vulnerable to relapse. And so the solution is to reintroduce the normal microbes back. This idea is not new, as Neil said. I'm not going to mention Chinese medicine because they were thousands of years ahead, but at least in the modern era of antibiotics, it was really first attributed to, in medicine, not veterinary science necessarily, but in medicine, to Dr. Ben Eisman, who was in Colorado at the time. And I was lucky enough to get into this field early enough to capture a statement from him, which I think is, which I always think about as we struggle with different frontiers in this field. So he said in his email, in the early days of oral antibiotics, we were plagued by frequent diarrhea in our patients, due presumably to killing off intestinal bacteria. I was chief of surgery at the VA, I should say in his 30s, and simplistically considered merely reintroducing normal organisms to counter their absence. Those were days when if one had an idea, we simply tried it. It seems to work. I wrote it up, made a small splash. Best wishes. He continued being active all the way until the very end, and passed away only a couple weeks after he was not able to attend grand rounds in his surgery department. And perhaps considered his contribution to this field a small footnote to his otherwise illustrious career in other parts. But everything he says here is still very relevant. And so back to this case, I thought, okay, if we're gonna do this, then we should do some science. And it's gonna be exciting. And his was the early technology. So who was gonna be the donor? Well, at the time, the state of the art was, well, you invite the intimate partner. That's the best donor, perhaps because this particular individual has shared all the microbes with the patient, and so you don't have to worry so much about infectious transmission. Obviously, this person should not have antibiotics, should be healthy, should be an adult who can give consent. That was pretty much it. And here you see some basic technology instruments. This was not the actual blender. It has a critical part. Here's the O-ring. I was so smart. I got a blender from the lab that was metal, and I could autoclave it, but it was missing the O-ring. That was a disaster that I was not going to repeat the second time. But that's, and also as a consequence of that experience, you see a face shield was introduced subsequently. All right, and additional ingredients is that at the time you had to have determination and courage. And what Neil's cartoon from South Park didn't capture was the intense smell that I could not anticipate. When you hit the blend button on that blender and you don't have an O-ring, it could literally knock you over. Suddenly the surface area of this material is 1,000 times greater, and there was no shield, there was no biological cabinet. It took nurses then the rest of the afternoon running around with air fresheners to clear out what happened. The result, though, we did measure what happened. And Neil showed this before. We had no funding at the time, but we found a collaborator in Sweden who did this rather primitive assay where you isolate DNA and then you subject it to degradation with enzymes and you get kind of a barcode. And you can see that before there was a certain kind of barcode in the patient. The middle column is the donor, who was the patient's husband of 40-some years. And then about 2 weeks later, you can see it looks pretty much like the donor. That was ultimately published in 2010 in a fairly low-tier gastroenterological journal because it was just an N of 1. But it was picked up by the New York Times. More importantly, the patient literally felt after 15 months of suffering, she said, while still in the waiting room, I feel different. And she was cured. This was picked up by the New York Times. And we were heroes at the time for the microbiome field, which was exploding because new technologies allowed looking at this frontier, which was not possible before. Prior to 2000s, microbiologists had to grow individual microbes in the lab, and the fact was that you could grow only 1% or so of what's there because you didn't know how to grow the other ones. Plus, they liked each other, so they had to be in a community. But with DNA sequencing technologies and computational techniques, now we could look at the whole microbiome. So that field was exploding concurrently, and here was an example where we did something and cured a patient and demonstrated this is important. And for the next year or so, I actually didn't do anything because N of 1 was not science, but N of 2 was science for the IRB, and it took me a year to get IRB approval to do another case. And then after the next year, I accumulated maybe about 10 cases of recurrent C. diff infection. And I was really unhappy with the approach of the trying to find a directed donor, whether it's intimate partner or a friend or a relative or a neighbor, because patients were just not able to bring the healthy subjects that would seem most desirable. There would be, here's my husband, and he had gastric bypass surgery, and he still weighs 300 pounds, and he has diabetes, and I'm thinking, oh God, am I transferring diabetes with this procedure or what? So I decided it was our responsibility to build a donor program, and ultimately we built an entire manufacturing program that took a number of years. It consists of 2 parts. One is that we have a donor program where we select the donors, and it's very rigorous. They have to be absolutely healthy, obviously no antibiotic history. They get tested for infectious disease, but they also have to be healthy in other ways because we still don't have enough knowledge to look at somebody's microbiome and say, this is really good stuff. So we go by clinical criteria. So these donors are like Greek gods. They're lean, they're beautiful, they have no autoimmunity, they have no metabolic syndrome, they have nice biochemical labs, no neurologic, no neurodevelopmental disorders, et cetera. And then the second part is the actual manufacturing. And as many of you know, in the real world, when you're embarking on manufacturing, you need to know that every step of the way is documented, is done the same way as it's written in the protocol. And somebody independent from quality assurance can go over and say, yes, you did it in these steps. So it's done in a university facility that's dedicated to different drug manufacturers, whether it's biologics or small molecules, and we have a suite there as well. It's standardized. The doses that we manufacture are standardized in terms of number of bacteria, not grams of stool. And in fact, we do know how many bacteria is necessary in order to get the optimal result at least in treatment of C. diff patients. It's 1 × 10⁻11th when you start losing the efficacy. And the fact is that there is about a 10-fold variation per gram of stool between individual donors, and sometimes even the same individual between different donations. We can track the units. We save aliquots. That means we can test for pathogens later. And the material is not released until all the testing is done on the donor as well as the donor material. Critical to this whole thing was the ability to preserve the material that's manufactured. It had to be frozen and still remain viable. So we introduced glycerol as the cryopreservative. This was truly a critical step because now you could test for things like HIV 2 weeks after the donation. And that would assure you that the donor did not pick up HIV on the day of donation or something like that. This is what FDA really wanted to see. That's what our initial unit was looked like in a cryo bag, concentrated. And you can see the little labels there, and we can cut them up, and these are little aliquots that could be stored. This could be administered in a variety of ways. It's liquid material. It can be thawed in about ½ an hour and ready to go. And it maintained the same clinical efficacy as fresh material, but it actually removed a lot of the fecal material. This is concentrated, mostly bacteria that are diluted in saline with glycerol. I wanted to show this slide because a lot of the readouts that people see is the catalogs of different bacteria, just the tables and tables. But that doesn't capture that these are microbial communities. And all these different microbes have relations with each other. They have different metabolic functions. They can work in concert and synergy. Sometimes they have competitive relationships. This is a very simplified map. It only shows some bacteria. It doesn't show viruses. It doesn't show fungi. It doesn't show the host and their relationships in all those directions. But what you see is that I think anybody, whatever your field is, whether it is communications, social media, you see this is a social network. Now look at this social network and look at the next social network, which is a C. diff patient. This is the patient with recurrent C. diff infection. It's literally a devastated microbial community. Now you have certain microbes are more dominating. If you think about it, if it's like a human community and you have all kinds of humans to make a community, sure, you can have some doctors, you can have some lawyers, you need some builders, you need some car mechanics, you need some hairdressers, etc. That's a community. Now imagine a community that's only made of lawyers. It's not very functional. And this is what happens with this transplant. So you go on the left, this is the donor, and the bottom is the patient that's treated before the treatment, and we call it intestinal microbiota transplant, since I really don't like the fecal word because it's being abused. And then after, about a week after, you have essentially a recovery of the entire community. You really, if you look at this, you'll understand you cannot achieve this with a single microprobiotic. This requires this complex kind of a therapeutic. All right, so back in 2013, this treatment has picked up a little momentum and got noticed by the FDA. And they got us together for a little workshop in Washington, DC. They listened to the scientists and they listened to the physicians for 2 years, and then they gave their prepared talk where they informed the entire community that anything that's used to cure, treat, mitigate, or prevent a disease is called a drug. And as such, this material meets the legal definition of a drug and a biological product. Now you could say, well, we're talking about transplants. In fact, that is the reason why we wanted the transplant word in the FMT, is to avoid this very thing. But they said, well, the law says transplant only applies to human cells and tissues and organs. This material is not human. Therefore, it's a drug, and it follows this paradigm. Now, I didn't particularly like this. I don't think anybody liked this. One of the reasons is this. That means if it's a drug, you have to make an application. This is just an amendment to an application that we already had in at the time, anticipating this. And the original application weighed, I put it on a scale, weighed 22 pounds and occupied a couple boxes. This is an amendment to our capsule product. But actually, at this time, I'm okay with it because we're in a different era. We're in the era of therapeutics development, and these are variable products, and it is reasonable to consider it as a drug. Problem is, we still have to invent or develop the appropriate science of pharmacology for this drug, because what we have so far, what we understand as pharmacokinetics, pharmacodynamics for regular drugs, has to be modified to accommodate this class of therapeutics. And here we're just talking about preparation, not how this drug behaves later, but that's something we study intensely in my program. So here's the product evolution in our program. In 2010, it was looking like basically a suspension that you can administer as a liquid, fresh material. By 2012, this is cryopreserved. You can thaw it out. Well, I already explained this. By 2015, we developed an encapsulated preparation of microbes. We had to work on this because it is not a trivial thing to go from one to another. There are additives called lyoprotectants, which allow you to preserve the viability of microbes during this process. And we went through a number of them, and you have to have a lyoprotectant that's not just as gentle to all the different microbes that are in there and doesn't kill them, but also in the end, you end up with material that you can powderize. Sometimes we would get a cake that looks like a granola bar, and there you go, not much you can do with it. So we discovered that trehalose actually was the best lyoprotectant that we had. We started using these capsules, and we see by 2018, the major innovation is the capsules became Swedish orange color, which is great because now you can do placebo-controlled studies. And this is the actual dose. This is all that's needed to cure a patient with recurrent C. diff infection. For disclosure, this was licensed to Finch Therapeutics. Their product, it looks a little different. They have 15 capsules. It's because they have a lot more trehalose in their product. We still use this. And this is our program. It's a Microbiota Therapeutics program at the University of Minnesota. We are mainly supported by philanthropy. And our main partner in that is Achieving Cures Together, which was started by actually a patient of mine who had ulcerative colitis. He was recruited to the University of Minnesota as a football player, but never got a chance to play because he developed severe ulcerative colitis. We actually tried to treat him with the microbes, made some success, but ultimately he lost his colon to a colectomy. He went through a pretty horrendous experience and developed a somewhat jaundiced view of our medicine, which in some ways is valid. He felt that there is not enough emphasis on cures. There's not enough collaboration. There's too much commercial interest in everything. And that kind of fit what our missions were. So I'm listing here some. So we do treat patients still with recurrent C. diff infections or fulminant C. diff infections. We treated about 800 people in our program. We have not charged a single one a penny for the microbiota transplant material. Our main mission, though, is to support academic clinical trials. Not all clinical trials, the fact is they're expensive. And drug companies put a lot of resources into them, of course. But their aim is not necessarily to push the frontier of science. Their aim is to get a product approved. And they can be very risk averse. And if you just look at C. diff trials alone, only about 15% of patients qualify for their trials or are able to participate because of the requirements of them. So we support a number of clinical trials, including that in C. diff, but also in autism and inflammatory bowel disease, cancer, in hepatic encephalopathy, academic trials that deal with sick patients where the industry perhaps is not quite ready to go. Importantly, our academic focus is on the pharmacology of these different therapeutics, understanding pharmacokinetics, that is, how does the drug behave in the body, as well as pharmacodynamics, how does it work? We do engage to some extent with regulatory policies, although it's very difficult to know what influence we can possibly have, and participate in events like this and trying to educate about what is emerging and what is possible. And I guess that is all I had.
Okay, Alex, thank you very much. We had a couple of questions about how you recruit the volunteers.
Building a donor program, is that what you mean by volunteers?
Yes.
Is not a trivial undertaking. So at first, we just put some posters up. That turned out to be quickly a mechanism that was obviously not the right one, because a lot of people feel that their microbiome is quite fascinating because they have all kinds of problems. And those, of course, are not the donors we want. So the advertising by posters was not it. I gave a talk in medical school to medical school students, first years, and one of them came and actually volunteered. She told me a very interesting thing in that she told her friends who are also medical students that she was going to do this, and they laughed at her. And that gave me some pause, and I realized I didn't do enough outreach to the public. We have not educated the public about this, and this is my fault. And so I started doing a little bit more of that. So initially, really, it was medical students that were the volunteers. Subsequently, I mean, there's our early medical students, undergraduate medical education, before they really have contact with patients. And then organically, what I discovered is that the donors themselves, as difficult as it may be to recruit the initial ones, then become the best recruiters for the subsequent. They tell their friends. And so at this time, we have probably over 100 applications on the shelf that we're ready to reach into in order to, to screen more donors. It's a very different program right now. Our donors get microbiome testing once a month. We do look for rational ways to select donors for different diseases. We do a lot of research on the normal population. And of course, the COVID has introduced additional levels of screening and such.
There was a question on how much you collect at a donation.
It's variable. So part of being a donor is feasibility. Not everybody is able to donate on demand. So in our protocol, you have to come to a supervised bathroom, GMP production. We don't just accept poop from outside as raw material. Who knows where it came from? And not everybody is going to come to the university in the middle of winter, park their car, and just donate material.
So do you restrict their diet, or do you recommend something in their diet the day before? Or
we gently encourage a healthier diet, which is more plants and less meat. We don't go beyond health advice. This is probably true, that more plants in our diet, more fiber is probably healthier. Sure. And we look for feasibility. So when the donor is recruited, at first we let them donate and we don't use their material. we let their circadian rhythm adjust to this, whether they're be able to do this or not. Not everybody's able to do this on demand. And so that they go for a couple of months, we just flush their stuff down the toilet. And we've learned that there is a learning curve to this, and the donors can adjust their rhythm, or they may just not be interested.
Right.
And
Now, do you screen for fungi and other viruses other than HIV?
we screen for many viruses, of course, besides HIV. Now we have to do COVID as well.
Right.
There's HCV, there's HBV, there's HAV. We do test for CMV and EBV because a lot of our patients, our recipients, are immunosuppressed because we do take up higher risk trials. And so, yes, they're also tested for CMV. And
Yeah,
EBV.
CMV could be a real danger if it was viable and got into immunosuppressed patients. I did have a question early on. You mentioned Dr. Eisman. Somebody wanted to know if he ever considered probiotics in giving it, or what kind of samples did he actually use?
The mythology is that he used surgery residents as the donors.
Okay.
In fact, besides there was a little flurry of activity in this field in late 1950s, early 1960s. It happened at the University of Minnesota as well, with Dr. Wangenstein here. And surgery residents were used in Minnesota. I know that for a fact. The competing drug at the time for this pseudomembranous enterocolitis was vancomycin, which came on the market in 1959. So somehow that became a very good option for this condition for a number of decades before C. diff really evolved into a much more difficult and toxic microbe.
And I had a question. Have you ever monitored or measured the oral microbiome in those donors that were the best to see if you could somehow develop a screen if there was a commonality among the best donors in organisms in their oral microbiome?
We haven't looked at the oral microbiome at all. We, as I said, we do sample them, we do study them. All these individuals have a fairly comparable microbial diversity in their stool. There is some variability in composition, as you might imagine. And we do things like measure what gases, for example, they make. There's a theory that ulcerative colitis might be driven in part by sulfur-containing gases like excessive amounts of hydrogen sulfide. So we have a trial, for example, ongoing, where we rationally pick the donors who have the lowest abundance of sulfate-reducing bacteria.
Very interesting. And a final question: have you ever treated MS patients?
Yes, with those with C. diff, of course.
Okay.
Not for MS specifically. And I don't have any data that we helped those patients with MS.
MS, but did you help them with their C. diff?
Yes. So,
Okay.
as Neil said, the success rates are fairly comparable across different practices, across different ways of doing this. It's about 90% if you do it with colonoscopy. These oral capsules have a success of about 80%, which gives you a little pause. What's wrong with those 20%? And something we're very interested in, and that's why we're studying the pharmacokinetics and things like that to see how we can optimize this.
Okay, Alex, thank you very much. I think we're going to go on.